Good morning, everybody. Thank you for joining us for this first research. We're saying webinar / Q&A. There'll be a little bit of learning that Armelle will take you through, and Armelle will introduce herself in a second. But also the function of this group and for you to join it is to ask questions. We want this to be very informal. We want you to feel updated on all the research. We want to get a guide from you on your level of understanding of research. And it's very, very informal, as I say. There are no silly questions. We really want to get to know each other. We want to build a relationship so that you guys feel comfortable with the direction that the research is going on. And like Armelle will allude to, your feedback and experiences of Myhre syndrome are only going to make this program successful. The patients and their families are crucial to our advancement of research. And we'll probably touch on this in more detail as we go. I've skipped ahead without introducing myself properly. My name is Kate Weirs, and I'm the Executive Director of the Myhre Syndrome Foundation. With me, you'll know I'll get excited and start talking a lot, so you'll have to just stop me. But I will hand over to Armelle now. And we're very fortunate. At the beginning of this year, you might have seen in some of our updates that we were fortunate to receive some anonymous funding that has really accelerated our opportunity for research. So we are incredibly excited at the foundation that we can now move forward with many of the projects that we had on our wish list. And part of that wish list was having someone like Armelle, who could be our independent scientific consultant and come in and just look at all of the things we're doing and then give us a plan to move forward. So without further ado, I will hand over to Armelle to introduce herself.
Hello. Do you see my screen?
Yes.
With MSF branch?
Yeah.
Okay, because I have two screens, so I want to make sure that I'm sharing the right one. So as Kate said, my name is Armelle Pindon. I have to check on the slide if it's really my name. So I'm not an MD, I'm a PhD. I'm a neuroscientist. I did my PhD at the INSERM in Paris, the equivalent of the NIH, at the Hospital La Salpêtrière in Paris, in the Institute of Myology, and I was working on the central nervous system. Then I also worked at the Guy's Hospital in London, and I worked on the injury of central nervous system. Then I went to the pharmaceutical industry in Janssen and Johnson & Johnson, where I still worked on neuropharmacology and metabolic disorders. I was doing cloning and expression and pharmacology around a certain type of receptor, among them serotonin receptors. And then for personal reasons, I went to rare disease world. My dad died from a rare disease, and my son has a rare disease too. And now I try to help the small organization or foundation or even big organization to drive their drug discovery program. So how to get to treatment. Thanks to my background in pharmaceutical industry, that's really something I try to communicate and orientate the strategy of those small rare disease organization towards that goal. So talking about today, so I call that the MSF branch, because I want you to understand that it's not a webinar where I'm going to give you a lesson and teach you something. I hope you will get something at the end of the branch, but the principle is really interaction. So you ask me questions. The first part, the 10-15 minutes, I'm going to explain you the update of what the foundation is doing and the strategy, and I will give you the tools so that you can understand that and you can understand your disease. So it's not a medical consultancy. I'm not going to give you advice on if you should be on losartan or not, or these kind of things. I want you to have the tools to understand what's happening in your kid's body or your body yourself, and want you to understand why we do certain things. So I will start today with just don't worry, it's a lot on the thing, but I just want you to understand that if you want to go to a treatment, there are different phases to get to a treatment that we need to go through. Everybody has to go through those. So you can say that there are, let's say, 3 parts. The first part is what we call preclinical studies. It means that it's all the study for treatment, everything you have to do before testing the treatment on humans and on patients. Then the second phase is called clinical studies, and that's where you test and develop the treatments, testing them on humans, on patients. And then there's the third part where when you have the luck to have a treatment that is accepted and on the market, you still study the treatment while it's on the market. And you need to consider if the patient can afford it, is it available to everyone? So that's the third part. But for the first 2 parts, it's really to test the treatment, and it's first tested not on human and then on human. So that's the preclinical and then the clinical studies. So the strategy that's the foundation is taking is work on every single aspect at the same time, so that you don't work first on the preclinical and then on the clinical and then on the rest. So you have to work on all the fronts at the same time, so that you can you gain time, you save time by working on all the different things. So what I define, in fact, as the 4 pillars that have to be developed in parallel. The first one is what I call the preclinical toolbox. So it's all the tools that researchers need to study a treatment, and I will explain that. So if we give them the tools, they don't have to produce the tool, so it saves time. The clinical readiness, it's also the same, creating the tools so that they can prepare for the clinical trials and save time, that they don't have to prepare those tools. We give them the tools. The third part is for us to start already some treatments, discovery. So we decided to go directly to the gene mutations, tackle the gene mutations, and target the fibrosis. I will explain later why we want to target the fibrosis. And the last part where we need to work on is all the interaction with the industry and the regulatory, so the FDA or the EMA in Europe. Really have a good, so that they know us, they know what we do, they know what tools we have developed, but the most important is that they know the patients, what's important for the patients, so that when an industry comes to them with, I have a treatment, they are going to ask, is it beneficial for the patients? But if they don't know what the patient thinks, they will not, it will be a problem for them. So we need to communicate with them. They need to know us and know us really, know the disease and what the patient think about the disease, what's their burden, what's their journey, so that they can say to the industry, yes, this treatment is appropriate, or no, it's not appropriate. That, not considering if it's efficacy or not, there is efficacy or not on the thing, but first they want to know if it's good for the patients. So going back to, so the first thing we do is develop I'm going to talk today more about the preclinical toolbox that we want to develop, and maybe a little bit of the treatment discovery. We'll see if we have time, because what I want really is you to ask me questions. So for the preclinical toolbox, so making the tools so that the scientists can spend time doing preparing the tools. So to test drugs, you can test them on cells. You can test them on organoids, so it's reproducing the organs, or you can test them in animal models. So usually everybody goes through cells first because it's easy to reproduce. The manipulation is easy, the experiment is easy. So that's the first thing they use. And then they can go either to organoids or to different animal models. The issue with animal models is that it doesn't really recapitulate the human physiology. So that's the reason why we go more towards organoids, human organoids, and the FDA agrees and pushes people to go more towards human organoids because we see so many studies that work really well in mouse and don't work in human because we are not mice. We can treat most of the disease in mouse, but not in human. So what did we choose to do at the foundation is to produce what we call iPSCs. So iPSCs means induced pluripotent stem cells. So those are cells that we take your blood, and in the blood there are some cells that are called PBMCs. So it's a mix of lymphocytes and other type of cells. And we are going to reprogram the cells to become stem cells. So from your blood as an adult or a child or whoever, we transform the cells that are in your blood into the cells that were, let's say, equivalent where you were an embryo. So cells that can become whatever they want. So that's the reason why they are called pluripotent. I'm sorry for my French accent. There are some words that are difficult to say. So they are pluripotent because they can become whatever. They can become a neuron. They can become a cardiomyocyte, so cells from the heart. They can become muscles, bones, whatever. So they are what we call stem cells. But they come from the patient. So they are really the stem cells. They have all the genetic material from the patient. They are the patient. And then we reprogram them to become, as I just said, whatever we want: bones, hearts, vessels, neurons, motor neurons, muscles. And so we choose which one. And from those cells, we can mix different type of cells and reproduce organs. So you are not going to reproduce a heart that's that big. You reproduce a mini organ. So for muscles, for example, you can have muscles that are that big, approximately, and you can see the contractions. So you can test different treatment on it. You can have intestine organoids. Well, liver is easy to do. So you have here all that's just a few, but there are way more than what is on the slide here. We are not going to do ADSCs for the moment. ADSC, in fact, it's coming from adults because as adults, we still have stem cells in our body. And we can take directly those stem cells if we go in the organ and take the stem cell in the organ and recreate the organ. But that's for very specific organs, and it's easier to create the iPSC so that we can differentiate in whatever we want. And if we want to go for neurons, for example, we are not going to go in your brain to take any. Well, anyway, it's very difficult to find a stem cell in the brain, but so we go through iPSCs. If you have any question, just interrupt me, scream, do something. I really would like to have that as an interaction. So is there any question about iPSCs? I'm not sure I was clear about what's
yeah. Do you mind just going over PCSCs again?
Yeah, so that's it's like maybe 10-15 years ago, or maybe more. No, I don't remember the date exactly. We discovered a way to take your cells and put them back at the stage of the cells where you were an embryon. So where you were just a kind of ball with only cells that look all the same, approximately all the same. You don't have cells that are neurons or a leg or heads. You are just a bunch of cells together. So we can take your cells from your blood, from your skin, and bring them back to that stage where you were an embryo. And then start again the process to differentiate them into a bone, into a muscle, into a brain. So that's very important, because then we can reproduce a part of you in a petri dish. So it's not going to be a mice that we modified that can look more or less, the organ can look more or less like a human organ, but it's not, because we are not mice. It's really taking the cells from a patient and recreate its organ. We cannot take a piece of your brain or your kid's brain to put in a petri dish and study it. But we can take some blood, induce them to be pluripotent stem cells, and then reprogram the cells to become neurons, or recreate a mini brain, or recreate the bone of your kid. And then when we are going to study that, compare to his brother or your mother or whoever is healthy, we are going to be able to see really the difference in the behavior of the organ and the cell, really from a patient compared to a healthy person, not from a zebrafish or a mice or cells that comes from whoever lineage you can imagine that are in the labs for the last 50 years. It's really the patient cells and the patient mini organ that we are going to compare to healthy organ. So that gives a lot of information about how the disease works, what's the problem. And it's a very good tool to test medication, to test treatment, either small molecules, so that's the way we call traditional medicine, or gene therapy or gene editing or ASO, whatever the treatment we want to try. If we try it directly on the mini organs from the patients or on the cells from the patients, that's a way better way to understand and be sure that if we have an effect, I mean, be sure, we are never sure in science, but it's a better way. We have more chance to be right, let's say, if you test it on the patient's cells in a petri dish before testing it in the kid, than in a mice or a cell line. Is that better? Or?
Oh, yeah, definitely. And what does ADSC stand for?
So that's adult stem
Okay.
cells.
Okay.
So that's what I was explaining, is that you can either take cells from somewhere and put it back at the stage of embryonic, or you can take directly stem cells that are still in your body. As adult, you still have stem cells. For example, in the muscle, you have cells that are named satellite cells that are stem cells, in fact, and can differentiate into muscle cells. So that's adult stem cells, but they are very specific to one organ. You have the same in the intestine. You have stem cells that you can go select, take, and then you can reproduce the organ from those stem cells. But if you take the stem cells from the intestine, you are not going to create a mini brain. You are going to create an intestine from the adult. Is that okay? Is there any other question? I don't know, Ashley, Laura, I don't really know you, so I don't know what's your level, if you can understand or not. No questions. Okay. So that's the first thing that the foundation did, is to create patient iPSCs. So they are going to, so we use the CRO A CRO is the contract research organization. So that's it's a company, it's a private company that does specific things. It's their job, so they do that all day long. They know what they do, and they are good to produce tools. They are efficient, they are fast. So in August, end of August, beginning of September, we should have already the iPSCs that we can give to researcher to start studying them. So that's the first tool. The second tool that we are going to have and to develop is really choose what assay. So an assay. It's the name to say how to measure something in biology. So if you want to measure the effect of a treatment, of a drug, you need to measure it, you need to use an assay. And in Myhre syndrome, we need first to determine what's the most important assay and the most relevant assay. What should we measure to estimate if a molecule, if a treatment is going to be efficient for Myhre syndrome? So that's an assay. And to determine which type of assay we want to have to test the drugs, we need to really study the different cells coming from the patient. So the iPSCs that we are going to transform again in neurons, in bones, in intestine, or in vessels. So all these kind of different models, we are going to study them so that we can know how we are going to measure a treatment in the preclinical phase.
What did the I stand for in the IPS?
Induced.
Okay.
We induce the pluripotent stem cells because we don't take the adult stem cells, we take the blood cells. So they are not stem cells in your blood, they are lymphocytes, they are other type of cells. So they have a function already. So to put them back at the stage of stem cell, we say that we induce them to be stem cells. So that's I.
And is the plan to develop several or like different, like liver and heart and muscle, or as many as possible, or just one at a time? Yes. Or
No. We the idea is to develop them in parallel. So we are having some meetings with the different scientists in Myhre to determine, in fact, to agree all together what would be the best one. So everybody agreed on cardiomyocytes and chondrocytes. Chondrocytes are the cells from the bones. I'm still discussing with some scientists about what would be the best for the vessels. So it should be a mix of different cell type that we are going to mix together and try to recreate a good model. But yeah, we are having this discussion, and in the RFP, so the proposal that we ask people to come with, I'm talking to the different applicants, and I try to push them to differentiate the iPSC we are going to give them and characterize them. So really compare them to healthy cells from the same family, that's the best, and see what are the difference.
Amel, question from me, just to tell everybody, so once the researchers have access to these iPSCs and they want to work on them, what are the types of work that we're going to want to see, or what are the types of work that they're going to be doing? So you talked about that they might test them for small molecules and compounds, so drug discovery. What else
That's
might they be
later.
doing? Yeah, what else might they be doing
So-
with these at the beginning, and then how they progress to that? If we can take people through that process.
Yeah. So the first thing is to decide what they want to differentiate. So they have the iPSCs, for example, Valérie Cormier-Der in France is specialized on all the bones and skeleton in Myhre. Well, she does other things, but she's really interested in that. So she wants to have the iPSCs, differentiate them in chondrocytes, osteoblast, osteoclast, make them grow and compare them to the healthy patients. So that's what we call characterization, so that we will understand for a Myhre patient, how do their bones behave differently than the healthy patient, the healthy control. So the characterization is the first step. And when you have understood the difference between a Myhre patient cell and the healthy control cell, then you can have a target, so you know what you have to address as the issue, and you know what to measure. So the first thing is the characterization. And I'm not going to ask one person to differentiate into 3 or 4 different cell type. Every lab has its specialty. You will have some labs that are specialized in neurons, for example, and they will do all the electrophysiology, but they are not specialized in bones. So every lab will receive, competent lab. We are not going to give them to anyone that asks them, but if you have a really nice project and you are a specialist in one of the aspects of the disease, then we give you the cells and you do this characterization. So for example, you can differentiate them in neurons, and you are going to measure the potential, the electrical potential in the neuron compared to the healthy neuron. You are going to compare the way they extend the ends of the neurons to contact the others. I mean, there's a lot of different tests that we can do to characterize how the cells from the patient behave differently from the healthy control. And then from there, we can know what to measure, what assay to choose, and we can start testing molecules. But as long as we don't have the characteristics, so to know what's the difference between Myhre and healthy, we don't know what to measure. So we need the characterization first. When we know what to measure, then we can measure and test molecules or treatments, whatever the treatment is.
Mm-hmm. And how long, Amel, do those steps take? The $1,000 question.
Well, when you have the iPSCs, the differentiation can take up to 3 months, depending on the cell, type you want to have. So that's already there, and then you need maybe 2 or 3 months more to do all the characterization, because you need to think about when you thaw the cells, you freeze them, you grow them to do every experiment, so it takes time. It's not that the experiment takes 3 months, it's more than you need to grow the cells, you need to, so it's let's say that's between the differentiation and the different experiment that you have to do to characterize, it's a minimum of 6 months. What I wanted to show you also is
Oh, sorry, Amel, I think someone had a question.
Yeah,
Go, Laura.
Go ahead.
Yeah. Armel, if I understand right, it would be the organoids characterized or the iPSC cells, how do you say?
Say it in Italian if you want.
I am Spanish.
Oh, you're Spanish, sorry. I saw Laura, I thought it was Italian.
No,
I don't speak Spanish, I'm sorry.
If I understand well, the CRO will make
Yeah.
the characterization of these organoids and make it ready for any researcher to have access, or it will be the iPSCs?
First, we start with the iPSCs for the CRO.
Yeah.
And then some of the differentiation and characterization can be done in CROs and other will be done in academia. Because not all the CROs, for example, for bones, you don't have CROs that really know about chondrocytes and osteoblasts. But for neurons, you have CROs that can do the characterization. The thing is that for characterization, what we need to think about CROs versus academia is that the CRO, they are technicians. So they know how to do something, but they don't have the knowledge and the background of the disease. So if they face a challenge that is linked to the disease, they might interpret it as a technological challenge, but in fact it's due to the disease. So there's we need to have a really nice interaction between the CRO and scientist, or we need to go to academia for certain things. It's a balance. It's something that you cannot decide I'm going to do all the differentiation in CROs. You need to be smart and really evaluate when it's necessary and when it will not be efficient.
Okay, thank you.
So what I wanted to show you with this slide also is the characterization of the fibroblast in human and in mice. And in fact, people used to think that a fibroblast is a fibroblast. So you used to take skin samples and a punch in the skin and say, well, this is the fibroblast. And now we know that the skin fibroblasts are not the same as the one in the lung, as the one in the heart. Just in the heart, human heart, there are 7 different types of fibroblasts. So Myhre disease really has a problem with the fibroblast. We have fibrosis, so that's a very important part. And if you compare not only the fibroblast in the human that are different from the skin, from the heart, from the lung, but the fibroblast from a mouse to a human, it's totally different. So that's also one of the reasons when you test drug in a mouse for fibrosis, it's very difficult to find a treatment that will be efficient in mice and then in human, because we don't have the same cells. So that's also one of the reasons why iPSCs, in fact, are becoming more and more important, because we realize that there is, oops, that's not for you, not for now. So that's the reason why the FDA pushes towards using iPSCs and patient organoids rather than mice, because we have so many clinical trials that cost a fortune, like $1 million, years of investigation, and at the end we see that it's not efficient. It was a fantastic drug in mice or even in monkey, well, usually in mice, and then when you go to human, it doesn't work. Still, you need to have animal models, because when you have a drug that you see is efficient and works in your organoid, you still need to know how the drug is going to be absorbed, how the drug is going to go everywhere in your body. So that's what we call the ADMET tox. So it's the administration, the distribution, the elimination of the drug, and the toxicity in the whole body. You cannot see that in organoids because it's just an organ, it's not a whole animal. So that's the reason why we still use animal models, but it's more for the ADMET-tox part of the study. Do you have any questions on that? Okay, so I just wanted to mention that we have an RFP now at the foundation, asking for scientific team to have a project on gene editing. So the Myhre syndrome has, I don't want to use the word luck, but let's say a huge opportunity, because the mutations are very close one from the other, and they are almost always at the same place in the DNA. So if you can correct these two different places at the same time, then you can have a treatment for 100% of the patient. So the gene editing is in fact removing the defective part of the DNA where you have your mutation or your variant, we say now, and replace it by the right sequence. So do you have any questions on that? Does everybody know about CRISPR-Cas, prime editing, gene editing?
Sorry, it might be helpful just remind everybody about that people can reference, if they know on their genetic report, that the numbers we're talking about is 496 and 500.
Yeah.
And that refers to the position. Sorry, losing my voice. And the letters before and after have been about the amino acid exchange. Maybe just take a step back on like what's happened.
Okay. Yeah, so that's the reason why I wanted interaction, because I have no idea about your level in science. So so I can start from the beginning, is that you have chromosomes. On the chromosomes, there are some genes, and the chromosomes are made of DNA. And the DNA is a kind of code, like a big book of recipe to produce proteins and produce also RNA. So in this book and the recipe, you have a sequence of letters. There are only 4 letters, A, C, G, T. And if you have the wrong letter, that's what we call a mutation. Every chromosome has a number. So for Myhre, the chromosome is 18. Then the chromosome has, you can see here. So the chromosome has a short part, a part that is restricted like when you put a belt on your pants. That's the centromere. And then you have a long part. So that's the P and Q part. So that's sometimes you can see on your results for the variants, you are going to see 18Q, which means the long or the short part of the chromosome, and then a number that is going to be the letter in your gene, the number of the letter. So the first letter is number 1, and the last letter is the last number. So if you have so this 18Q, the number, and then a C, the C means cDNA. So it's going to be the location on your DNA. And the location can be C1498, for example. So that it means that in your gene, the SMAD4, on the 18 chromosome, at the DNA nucleotide, so that's the letter 1498, you have a change, and this change might be, don't remember by heart, but G bigger than A. It means that you have switched G by A, and that's the error in your DNA. Instead of having a G, you have an A, or instead of having a C, you have a T, or whatever. So that's the change that you have. Then you have another number, which is about the protein. So something you need to know is that you have in your chromosomes, you have the DNA, so all the code, and this code is to build, for your cell to build proteins. So you will have the DNA, it's going to be transcribed into another messenger because the DNA cannot get out of the nucleus. So in a cell, you have the cell, it's kind of balloon, and in the cell, you have another smaller balloon, which is the nucleus, where you have all your chromosomes. They always stay inside the nucleus. They cannot go out. So to get this information from your chromosome to a protein in the cytosol or in the branch of your cells that are going to touch another cell, you need somehow this information to go into the rest of the cell, outside of the nucleus. So for that, you have a messenger, which is called an RNA. So it's not anymore DNA, it's RNA. So if you have the DNA that has a mutation, so a mistake, you have a G instead of a or A instead of a G. When you reproduce and take the messenger, is going to read that and take the exact message. So the messenger, the RNA, is going to have exactly the same error. And then this RNA is going to go outside of the nucleus in what we call the cytoplasm. And it's going to give this information to build the protein. But the mutation is still there. This information that is now on the RNA is coded by 3 letters by 3 letters. And at every sequence of 3 letters, so for example, GAT, you will have a small part of the protein, what is called an amino acid. So that's all the different balls that you put one after the other, and at the end you will have a protein. It's the building blocks of a protein is an amino acid. So that's what you see when you see P. So that's for protein in your variant results, P dot ILE, for example, 500 val. It means that in your protein, at the position 500, so the 500th amino acid, instead of being the amino acid called isoleucine, you are going to have a valine, because the code that you had not the right 3 letters, but had a change in the letters that became that code for another amino acid. So the P is for the position in your protein where the problem is. Explaining all that without slides, I'm not sure you followed anything.
No,
So
that was
next time,
amazing.
maybe I
Amazing.
can prepare the slides.
Thank you.
Because it's a lot of information just saying like that out loud without visual support. I can have the visual support for next time.
Yeah, and just
repeat that.
Just to remind everybody, in our handbook, I can send it round. We have a couple of pages on here that has that visual representation of that process that Amel just mentioned too.
So one of the treatment we choose to invest in is correcting this error in your DNA, and that's called gene editing. And if you correct both errors, if you produce a tool that can correct any error between two spaces, 2 locations, that's called a prime editing. So instead of correcting the errors one by one, we can correct with the same tool. We can use it to correct all of them. So it's a choice. We can go either to correct one by one, because there are not that many mutations in Myhre syndrome, or we can choose to develop a tool that corrects all of them at once. So that's, yeah, I think this is too complicated. That's the prime editing with the CRISPR-Cas, but I don't think you are. That's for another time. So I'm going to stop sharing and just answer your questions. I talked a lot and you did not. So we have 10 minutes more, and I really would like to know where you would like me to maybe give you more information, what you don't understand, what's your level. Just interact, whatever question you have. Do you know what's a drug? Do you know what's a treatment? Do you know, I don't know, just ask me. Hi, Lisa.
Hi, I was gonna wait a minute. I don't want to hog all the questions. If anybody else wants to go ahead.
You go, Lisa. You're fine.
And it might be, we might be too early in the process, but what do you guys think, maybe the name of the most promising possible treatment might be? I mean, you don't have to explain what CRISPR or CRISPR or whatever, but maybe what just the name is of what you guys think would be the most promising possible treatment.
Well, that's a $1 million question. So the logical way to go in terms of modifying DNA or messenger RNA, as I said, or protein, is the prime editing or the editing, let's say. That's the logical way. Another way that we are going to go in parallel at the same time is to try to find repurposing drugs, so drugs that are already on the market or drugs that are in development for other diseases. And as soon as we have characterized the cells, we are going to test also small molecules, especially for fibrosis, because that's the main problem. I mean, that's the life-threatening problem with Myhre, is coming originally from fibrosis and stenosis. So we are going to go also for treatment that will address specifically the fibrosis. Those kind of treatments would not be, if we think about it, might not be chronic treatments, might be treatment that we would use in case of emergency or when we see that there is a lot of fibrosis coming up. And the gene editing might be a treatment that is not for acute treatment, but well, if you correct, that's it. So we go for both of them at the same time. In fact, 3 of them, the gene editing, the fibrosis with a specific technology, a new technology that I will explain next time, and screening for small molecules, repurposing drugs. So that's the 3 ways we are going now. But telling you which one is going to succeed, that's I can't answer that. We can do our best and try the 3 of them, but
That's great that you're able to do all 3 ways, 3 possibilities. And in general, is it quicker to get, well, I guess we're not really, medication production is not in there, so we don't have to worry about that. It's more repurposing of drugs, because my question was going to be medication production quicker in Europe than in the US, but we don't really have to deal with that because it's repurposing of something that are current drugs.
no, and before thinking about drug production, we need to find a drug first.
Mhm.
That's the production will be for if we are lucky enough to have a drug, then we can think about production.